Suppose you are working onto a global development product, the product which is being developed for the global market, including the regulated, semi-regulated, and some other markets. Or in other words, you can consider or suppose that you are working onto a formulation for the US market, for the Europe market, Brazil market, ROW market. And you find that there are some differences in the quality requirements across the regions. The USP has some criteria for the dissolution specification, impurities, while BP has some other criteria. Then how you will address this? I will suggest that you need to go with the most stringent criteria. If USP is more stringent than other pharmacopoeias, then you should follow that. But it is not always possible and it is difficult. So the Pharmacopoeial harmonization came into existence. I welcome all the viewers to this topic on pharmacopoeial harmonization. Now suppose that you are working onto a product and the USP, International Pharmacopoeia, for example, let's say Chinese Pharmacopoeia and Japanese Pharmacopoeia, all have the same requirements and the same acceptance criteria. So it will be easy for the people who are working on the product. It will be easy for the regulators. It will be easy for the organizations or companies which are developing the formulations or working in that area. This is the importance of pharmacopoeial harmonization. Now we will see the pharmacopoeial harmonization in much detail. By definition, pharmacopoeial harmonization is the process of aligning and standardizing the quality requirements, test methods, and specifications for the medicines across different national and the regional pharmacopoeias. It ensures that medicine tested under the USP, PHUR, JP, BP, or other pharmacopoeias meet the equivalent standards without unnecessary duplication of the testing or reformulation. So this is the main importance and requirement. For example, you are working onto a product, let's say tablet formulation, which is being developed for the US market and also for the Europe market. USP provides you some dissolution criteria with some parameters, while BP provides you some other criteria and the parameters. So it will be difficult for you to meet both. But if the criteria is same, if the testing parameters are same and specifications are same, then it will be easy and it will avoid reformulation. It will avoid unnecessary duplication of the testing. It will save your time as well. Here is the requirement for pharmacopoeial harmonization. So why pharmacopoeial harmonization is needed? Different countries have different pharmacopoeial standards, which creates challenges for the global pharma companies and the professionals. It leads to duplicate testing, higher costs, and delays in the drug approval. It leads to time consumption in the repetitive testing and meeting the specifications. Harmonization facilitates global drug development, regulatory approvals, and the trade. Also, this harmonization supports and helps ensures the patient safety with consistent quality worldwide. Let's say, for example, USP monograph provides the dissolution with 20 minutes or 30 minutes as a Q point, and BP provides 45 minutes as the Q point. So if the product is developed as per BP standards and if it is not meeting the USP standards, it will lead to reformulation. But if both the pharmacopoeias have the same requirement, it will save the time and the resources. Key initiatives in the pharmacopoeial harmonization. So PDG group is very important to consider here. Pharmacopoeial discussion group, it was established in 1989, and members of this PDG group are USA with USP, Japan with JP, and Europe with PHUR. International Pharmacopoeia from WHO and Chinese Pharmacopoeia from China participate as the observers. The focus of PDG is on harmonizing the general chapters and the excipient monographs. For example, the general chapters like dissolution, 711 USP, and sterility. Then ICH Q4B guideline, it provides a framework for regulatory acceptance of the harmonized pharmacopoeial texts. Example, if a dissolution test is harmonized, results using USP, JP or PHUR are considered interchangeable. You might be knowing that many of the times USP guideline come with a revision or new ICH guideline. It is ICH guideline and then US FDA adopts the same. So it will be easy for all the professional and the organizations to adopt it. Third is the WHO's rule. International Pharmacopoeia promotes harmonization for developing countries and it is published by the WHO. It supports global access to quality medicines, especially in public health programs for different diseases like HIV, TB and malaria. What are the benefits of the pharmacopoeial harmonization? So as the testings will be similar, the acceptance criteria will be similar, so it will reduce duplication of testing in global pharma and it facilitates faster regulatory approval, it supports international trade and exports, and pharmacopoeial harmonization ensures consistent quality standards worldwide. It strengthens patient safety and global public health. So these are the some of the important uh, advantages. Then challenges. Some challenges are there as the different pharmacopoeias have different requirements. So challenges are differences in the regulatory systems and the legal requirements. Then variations in the analytical methods and the acceptance criteria. These may be the challenges. Then need for continuous updates as science and technology evolve. So for example, if one pharmacopoeia uses the dissolution testing by UV and if another one is using by HPLC, then it is very challenging or it becomes challenging to update the all pharmacopoeias. That means it is challenging to have the harmonization. In short, pharmacopoeial harmonization is making sure that if a medicine passes a test in USP, PHUR or JP, it is accepted globally without retesting and thus streamlining drug development and ensuring consistent quality across the region or across the borders. Now coming to the examples for the harmonized and non-harmonized chapters. Comparison table is included here for the harmonized versus non-harmonized pharmacopoeial chapters under the Pharmacopoeial Discussion Group (EDG) framework with USP, JP and PHUR. So category is the examples of harmonized chapters and non-harmonized chapters. General test for the quality like dissolution, disintegration, uniformity of dosage units, sterility test, microbial limit test, bacterial endotoxin test, particulate contamination. These all are the harmonized chapters. Non-harmonized chapters are powder flow properties, water determination by Karl Fischer, thermal analysis, particle size distribution by laser diffraction. These are the examples of non-harmonized chapters. Then analytical methods. So chromatography, that is PLC, HPLC, infrared absorption spectroscopy, pH measurement, conductivity, all are the harmonized analytical methods. Some examples for non-harmonized analytical methods are near IR, spectroscopy, mass spectroscopy, and Raman spectroscopy. Examples of microbiology, that is sterility, microbial enumeration, test for the specified organisms. These are harmonized. Rapid microbial methods, mycoplasma testing are non-harmonized. Dosage form test, dissolution, disintegration, uniformity of dosage units, these are harmonized. And non-harmonized dosage forms like inhalation test, product testing, then the transdermal patch testing, these are coming under the non-harmonized chapters. I suggest you to go through the recent understanding and recent updation on these examples so that you will have the current knowledge. Excipient monographs are also harmonized for MCC, magnesium stearate, lactose, starch, and these are non-harmonized for some co-processed novel excipients like co-processed excipients, PEG derivatives, and lipid-based excipients. Similar is the case for the biologics like bacterial endotoxin test and sterility. These are harmonized, and biologics potency assays are non-harmonized. Harmonized chapters are accepted interchangeably in the USP, JP, PHUR, and these are as per the ICH Q4B guideline. Non-harmonized chapters are country specific and companies are required to comply separately for these non-harmonized requirements. Harmonization mainly covers the general test methods and common excipient monograph, not all the drug substances. And if the monographs are non-harmonized or if the requirements are non-harmonized, then the manufacturers, the companies, organizations are required to comply those requirements. Then biologicals, inhalation products and advanced therapies are still under the non-harmonized territory. This is regarding the pharmacopoeial harmonization. Thank you for watching the video. Let's meet soon with another informative video. Thank you.